For decades, the search for habitable exoplanets has been guided by a singular, optimistic vision. Finding a world lush with vegetation and liquid water, a second Earth. Organizations like NASA and SpaceX have invested billions in technologies designed to bridge the vast distances to these potential sanctuaries. However, a deeper analysis of biological imperatives suggests that discovering a thriving biosphere might not be the salvation of humanity, but rather a profound biological threat. To understand why this matters, we must look back to July 1969, when the Apollo 11 astronauts returned from the moon. They did not simply walk off the recovery ship. They were immediately sealed inside the mobile quarantine facility. NASA engineers and biologists understood a critical truth that is often overlooked in modern discourse. Planetary exploration is an exchange of biological data, and in biology, a pristine environment is often a battlefield. In this analysis, we will examine the often ignored risks of visiting a living world. First, we will assess the probability of cross-species viral compatibility, analyzing whether alien pathogens could biologically interact with human physiology. Second, we will investigate the threat of chemical toxicity and chirality, where even basic organic compounds could act as potent poisons. Finally, we will address the ethical and practical dilemma of forward contamination, questioning whether our own microbiome makes us too dangerous to ever set foot on a living world. Drawing on principles of evolutionary biology and current planetary protection protocols from NASA, we will explore why the safest destination for humanity might ironically be a dead world. Before we analyze the specific biological mechanisms, if you appreciate this engineering-focused approach to space exploration, please take a moment to like this video. It helps support deep dive content. To fully grasp the magnitude of the biological risk, we must contextualize our current search for life within the framework of aerospace history and evolutionary theory. The concept that contact with an alien biosphere could result in catastrophe is not merely science fiction. It is a foundational concern of astrobiology. In 1898, H.G. Wells published The War of the Worlds, introducing a concept that remains scientifically relevant today. In his narrative, the Martian invaders were not defeated by human weapons, but by the humblest things upon the earth: bacteria. Wells illustrated a crucial biological reality: that an immune system evolved in isolation is defenseless against novel pathogens. Today, astrobiologists refer to this as the War of the Worlds reversal. The question is no longer just about Martians invading Earth, but what happens when human astronauts become the invaders on a planet like Kepler 186f. This theoretical risk drove the engineering decisions behind the Apollo program. Between 1969 and 1971, NASA implemented strict planetary protection protocols. Upon splashing down in the Pacific, the crews of Apollo 11, 12, and 14 were required to wear biological isolation garments and were transferred immediately to the mobile quarantine facility. NASA's concern was back contamination, the possibility of bringing lunar microorganisms back to Earth. While the moon proved to be sterile, the protocol established a precedent. We cannot assume a new environment is benign. The scientific principle driving this concern is convergent evolution. This is the observation that distinct species will independently evolve similar traits to adapt to similar environments. For example, the camera-style eye has evolved independently on Earth approximately 40 different times. This leads to a critical engineering and biological trade-off. We actively search for Goldilocks planets, worlds located in the habitable zone where liquid water can exist. However, the conditions that allow for liquid water and lush forests are the exact same conditions that support predation, competition, and pathogens. If a planet is capable of sustaining complex human life, it is, by definition, capable of sustaining complex threats to human life. In the next section, we will analyze the mechanics of these threats, specifically focusing on the debate surrounding viral compatibility. If you want to stay updated on these technical deep dives into space exploration, please consider subscribing to our channel. To evaluate the safety of an inhabited exoplanet, we must first analyze the potential for biological interaction. The primary question facing astrobiologists is essentially a debate between two evolutionary theories: the lock and key hypothesis versus convergent evolution. Virology on Earth operates on high specificity. A virus acts as a key that requires a specific molecular lock or receptor on the surface of a cell to enter and replicate. Because human DNA is the product of billions of years of specific terrestrial evolution, the prevailing optimism among some scientists is that alien viruses would be effectively blind to human physiology. Our genetic makeup would simply be too alien for them to hijack. However, this view is challenged by the principle of convergent evolution. As previously noted, functional structures like the eye have evolved independently dozens of times on Earth because they solve a universal physics problem. If the biological solutions to energy consumption and replication are universal, viral structures may also be convergent. The danger is not necessarily a predator we can see, but a microscopic compatibility we cannot predict. Even if an alien pathogen cannot infect human cells directly, a secondary, perhaps more insidious threat exists: the destabilization of the human microbiome. The human body acts as a host to approximately 30 trillion bacteria, which are essential for digestion and immune function. In nature, bacteriophages are viruses that specifically target bacteria. If an alien virus were to ignore human cells but target our gut flora, the result would be catastrophic. The destruction of the microbiome would render an astronaut incapable of processing nutrients. In a clinical sense, one could die of starvation while having a full stomach, simply because the biological machinery required to digest food has been neutralized. Beyond active biological agents, we must consider the passive environment. The distinction here is critical. A predator attacks, but the environment itself can be toxic. Consider the pollination cycles of a hypothetical biosphere. On Earth, pollen triggers histamine reactions. On an alien world, airborne particulate matter could contain heavy metals or arsenic-based compounds, turning every breath into a toxic event. More fundamentally, we face the engineering hurdle of chirality, or molecular handedness. Organic molecules have a specific orientation, left-handed or right-handed. Life on Earth is built almost exclusively on left-handed amino acids. If an alien biosphere evolved using right-handed molecules, the local flora would be biologically incompatible with human digestion. Consuming such matter wouldn't just provide zero nutrition, it could lead to the bioaccumulation of unprocessed molecules in the liver and kidneys, similar to plastic toxicity. Finally, we must address the risk of proteopathy, specifically prions. Prions are misfolded proteins that contain no DNA or RNA, yet they are infectious. They operate by inducing other proteins to misfold, causing a chain reaction of tissue damage, most notably seen in bovine spongiform encephalopathy, mad cow disease. The danger of an alien protein is that it does not need to be alive to be lethal. Simple physical contact or ingestion could trigger a cascading failure in human neurological systems. Unlike a virus, which can be targeted by the immune system, a prion is a structural flaw, a mechanical failure at the molecular level against which we currently have few defenses. If you find this analysis of biological trade-offs valuable, please click the like button. It helps us gauge interest in technical content. While the prospect of alien pathogens is daunting, a comprehensive risk assessment reveals a perhaps more probable scenario. The danger comes not from the alien world, but from us. This concept is known in aerospace engineering as forward contamination. The human body is not a singular organism. It is a walking ecosystem containing approximately 30 trillion bacteria. Even with the most rigorous sterilization procedures for our spacecraft, the astronauts themselves act as biological carriers. We must consider the historical precedent of the Columbian Exchange on Earth. When isolated civilizations made contact, the introduction of novel pathogens like smallpox devastated populations that lacked acquired immunity. On an exoplanetary scale, this dynamic could be exponentially more destructive. Earth's bacteria are battle-hardened survivors of billions of years of fierce evolutionary competition. If we were to introduce these aggressive terrestrial microbes to a biosphere that has evolved in isolation, perhaps one that is slower or less competitive, the result could be rapid ecological collapse. A single breach in containment could allow Earth-based mold or bacteria to consume native alien flora, effectively rewriting the planet's biological history before we have a chance to study it. This raises a profound ethical question. Do we have the right to explore a living world if our mere presence could result in the extinction of its native life? Given these biological incompatibilities, toxicity on one side and contamination risks on the other, the engineering reality of colonizing a habitable zone planet is starkly different from the artistic renderings of open-air settlements. If we cannot breathe the air due to toxicity, and we cannot touch the soil due to contamination protocols, we cannot live off the land. Instead, human habitation would require hermetically sealed, closed-loop environments. Residents would effectively live in a bubble civilization, observing the alien world through thick glass or camera feeds, permanently separated from the environment they traveled light years to reach. This aligns with NASA's current Office of Planetary Protection protocols. Currently, the strictest regulations apply to Category 5 missions, those targeting bodies of potential biological interest. Under these rules, finding a planet with extant life might legally and operationally ban us from landing on it, limiting our exploration to orbital observation to preserve the planetary environment. If you enjoy this detailed analysis of the ethics and engineering of space travel, please subscribe to the channel. We have examined the prospect of habitable exoplanets from multiple engineering and biological angles. What stands out is a stark contrast between our romanticized vision of space exploration and the biological reality. First, the biological risk is asymmetric. While the lock and key mechanism might protect us from some specific infections, the principle of convergent evolution suggests that alien pathogens could still target our fundamental biological systems or our microbiome. Second, the environment itself poses a chemical threat. Through mechanisms like chirality and toxicity, a lush alien forest could be as lethal to human physiology as a vacuum, requiring us to live in sealed isolation despite the presence of an atmosphere. Third, the ethical constraint is significant. Our own biological footprint creates a risk of forward contamination, potentially forcing us to quarantine entire worlds to prevent ecological genocide. The case for the dead world. Looking ahead, this analysis suggests a counterintuitive conclusion for the future of aerospace engineering. We spend billions searching for Earth 2.0, worlds teeming with life. However, the safest destination for humanity may not be a second Earth, but a dead world. A sterile planet, such as Mars or the Moon, presents no viral threats, no toxic pollen, and no native ecosystem to destroy. It is a blank canvas. While the engineering challenges of a dead world are physical, radiation, gravity, temperature, these are variables we can calculate and overcome with physics. The challenges of a living world are biological and evolutionary, variables that are unpredictable and potentially uncontrollable. Connection to opening. Returning to our opening premise, we often view the silence of the universe as a sign of loneliness. However, when we consider the biological imperatives of survival, that silence takes on a different meaning. In the search for a new home, a quiet, sterile world offers something a lush, living planet cannot: safety. If you found this conclusion thought-provoking, please like this video to help others find this content. Understanding these biological paradoxes gives us a more mature, realistic perspective on our cosmic neighborhood. It reminds us that while the universe is vast, the conditions that allow for our specific form of life are incredibly fragile. True exploration requires not just courage, but also the wisdom to recognize when a new world is best admired from a distance. I am curious to hear your perspective on the ethical dilemma we discussed. If we were to discover a planet with primitive life, simple moss or bacteria, but knew that our arrival would likely destroy it, do you believe we have the moral right to land? Or should we leave it untouched? Please share your thoughtful analysis in the comments section below. In our next analysis, we will move from biological threats to engineering solutions. We will explore the specific terraforming technologies proposed to turn dead worlds like Mars into habitable environments, examining the physics behind atmospheric processing. If you value this type of in-depth, unemotional analysis of space technology and science, I would appreciate your subscription. It allows us to continue producing high-quality documentaries for this community. Thank you for watching and for your time. Until next time, and